Good morning or good afternoon, depending on where you're joining us from today. I want to thank all of you for joining us for today's webinar presented by the National Institute of Neurological Disorders and Stroke, or NINDS. My name is Gabe Gonzalez, and I'm a health program specialist in the NINDS Division of Neuroscience, and I'll be your host today. Today's webinar is entitled The NIH Perspective on Common Data Elements. And the goal is to share how NIH is thinking about CDEs across the research pipeline, why they matter for data quality, interoperability, and sharing, and how they support the NIH's broader mission around rigor and reproducibility. Before we get started, I want to quickly flag that this webinar is part of an ongoing series focused on practical data standards, especially common data elements, metadata, and interoperability. To make preclinical and clinical data sets more reusable and comparable. We're actively looking for future speakers and panelists. So if you're developing CDEs, using them in a study, building tools or repositories that depend on standardized data, or you've learned hard lessons implementing standards in real workflows, this is exactly the kind of perspective we want to feature. If you're interested, please email us directly. We welcome short talks, case studies, demos, what I wish I've known, implementation stories, and we're aiming for a mix of NIH, academic, and community perspectives. Also, please take a moment to register for our Idea Scale campaign. It's a community forum to submit ideas, comment during public review of preclinical CDEs, and share implementation challenges. And your feedback is essential to help make preclinical CDEs more findable, accessible, interoperable, and reusable. And finally, before I introduce our speakers, we'll have just a few housekeeping items. First, today's webinar is being recorded, and the recording will be available on the NINDS YouTube channel, hopefully within 2 weeks. We'll place the link in the chat so you can access it and share it with your colleagues. A live transcript of the webinar will also be available. And finally, the chat will be turned off for this webinar, but we do invite your comments and questions. You should see a Q&A box on your screen. Please feel free to type your questions in the Q&A box at any time. And if your question is directed to a specific speaker, please indicate that. We'll compile the questions and address as many as we can during the discussion panel at the end of the event. And with that, I'd like to introduce the NINDS Preclinical CDE co-chairs and our panel of speakers. Our co-chairs are Dr. Hiba Awad and Dr. Ben Chern. Dr. Awad is a program director in the NINDS Division of Neuroscience, where she manages one of the division's 2 TBI-related portfolios. Dr. Awad previously studied mechanisms and pharmacotherapies in rodent models and TBI, and currently leads and supports the NINDS neurotrauma-related efforts, including preclinical, data standards, and CDE activities. Dr. Chern is a program director in the NINDS Division of Neuroscience. He supports research on acquired epilepsies, focusing on circuit mechanisms and seizure initiation, detection, and prediction, as well as refractory seizures and status epilepticus and related comorbidities across the lifespan, including post-traumatic and stroke-induced seizures and aging-related cognitive impairment. Dr. Chern is also Dr. Chern does serve as a lead on several initiatives, including the preclinical data standards and CDE activities. Our speakers for today's webinar are Dr. Pat Belgowan, Dr. Sinyi Steve Sang, and Dr. Carolina Mendoza Puccini. Dr. Belgowan joined the NINDS in 2014 as a program director, and he currently serves as the acting associate director for the NINDS Division of Neuroscience. Dr. Belgowan is trained in behavioral neuroscience and functional imaging with prior work on concussion outcomes. He is also experienced in cross-IC and interagency neurotrauma and informatics initiatives, including FitBur. Dr. Sang is a clinical informatics program officer in the NIH Office of Data Science and Data Science Strategy. He promotes research use of standards such as FHIR or FHIR and CDEs, and he leads training and education for reproducible biomedical data science. Dr. Sang was previously a program officer at NIAID, overseeing data science projects. And last but certainly not least, we have Dr. Mendoza Puccini, who currently serves as a program director in the NINDS Division of Clinical Research, where she leads the NINDS CDE program and the Hill CDE project, providing strategic oversight for CDE development, governance, and implementation to advance data harmonization, interoperability, and high-quality data sharing across neurological research. And with that, I'll go ahead and hand it over to Dr. Patrick Belgowan, our first speaker. Pat, it's all yours.
All right, thanks, Gabe. And I want to just thank all the organizers in the neurotrauma webinar series. That's Claudio, who's not. I don't know if he's able to make it today, but thank you to Claudio and Gabe who are leading this incredibly important series. This is a new way for, at least at NINDS, for us to reach out to the community and talk to you. So as Gabe said, I'm currently the acting associate director for the Division of Neuroscience, but prior to that, I was a program director for TBI. And what I'm going to talk to you about today is when I started as TBI program director, there had been this one task of preclinical CDEs had started. And having just started myself, I couldn't figure out why anyone would want preclinical CDEs and what would they be useful for. And so what I'm going to talk to you about today is sort of how my thoughts have emerged and how I've come to the place where I think that, you know, the future is in the preclinical CDEs, making sure that they match up with the clinical CDEs. So if you want to go to the next slide, please. So part of that is reframing, part of all of this, and I think part of what we need to do as a group, as a community, is to start to reframe the purpose, in particular, the way we use data. So in the past, and when I had a lab, the data I always felt was mine, but I think we've moved past that, and now we have to go even further. So we have to look at how do we get the money to collect the data? And that comes from the taxpayers. So we need to be able to provide the taxpayers with a product that's more than just the papers you produce. And that product, I think, is a national resource in biomedical research data, so that the data you collect and now the policies that NIH has require you to put it in a data repository somewhere. Which is important. It's a way for taxpayers to see how their investment is being used, and it allows it to be even more impactful. And I'll get to that in just a minute. But I think it's really important to understand that to optimize this national biomedical data resource, we need to do a couple things. And you're going to hear me say sort of the same things over and over. But one is building data standards from the start. From the start of your experiment. It should be something you're thinking about when you begin your experiment, when you design your experiment, and all the way through your experiment. You should require within your field a high quality metadata. And I'll show you in a little bit how you can assess that. We should follow the fair data practices and hopefully make your data machine readable so that it's easy to find and easy to use. And you should also be thinking about secondary use, not just your publications, right? So, for instance, for your data to be more usable, maybe you want to align some of your outcome measures with other studies. Rather than doing the Belgowan test of intelligence, maybe you want to do the WASAR, right? Maybe you want to do the water maze, the same as another group does the water maze, so that your data will be more easily combined for secondary use. And all of this, again, is to maximize the return for taxpayer investment. So I think in the last couple of years, we've really learned that it's important that the taxpayers understand what their money is going to, what the research does for them. And the more they do that, and the more they know, because we all know that it's really important, but the more they know that, the more they're going to be willing to invest and keep up our research enterprise. Next slide. So basically, for me, the message is that the data aren't the byproduct of research. I think we have to start seeing them as the resource, as the national resource that the community can use. NIH has provided a lot of infrastructure, but infrastructure alone is not enough. So here is a list of just NINDS's data repositories and biorepositories, and you can see it across a lot of disease areas, across a lot of different types of data, and so it's a very complex ecosystem that we work in, and so that complexity in and of itself of the data types really should be pushing you towards having common data standards and data structures. Next slide, please. So, as I said, each of you, as you start to prepare to execute your studies, first you designed the study, and I know you applied for a grant and got a grant, and congratulations on your grant. But then you start to collect the data, you make your discovery and report it. And now you need to put that data and those reports into repositories. The reports are often in fairly standard structures, right? The journals require you can't just write the paper any way you want. They require some standardization. Whereas the data itself, a lot of the repositories that you may be submitting to don't require structured data, and that is where we're losing some value for secondary use and rediscovery. Next, sorry. That was my bad. So what I'm promoting is that you look at common data structures and standards throughout the whole process, so that it's always in your mind, and it's always there to help us figure out how to reuse your data and combine your data in the future, so that it can have a bigger impact and lead to more discovery beyond what you may even want to, may even lead to more collaboration. So to do this, NIH has provided quite a few data tools and policies. So there's the GREI metadata scheme, which uses DataCite. I have a link there to the Zendo that will run you through the metadata scheme that they're recommending. And then recently, ODSS provided a NIH metadata completeness tool. So that allows you to assess the completeness of the metadata that you'll be submitting as a community that you've come together to submit. We've already looked at the data and biospecimen repositories in neurotrauma. We have TBI has had FITBR for quite a while for clinical data. Preclinical data, I'm assuming it must be almost a decade now that we've had ODC SCI and ODC TBI. And it's been more than a decade that we've actually had common data elements. And what we need to do is, what I'm hoping you'll do anyway, is to start using them more and more within the preclinical space. NIH also provides access to real-world platforms. It'll help you lead and understand AI readability and computer and cloud computing. Next. So the key takeaways, the key takeaways just are that standards should be in all the time. That harmonization enables cross-study comparability, and using that is the best and probably the best use of the taxpayer dollars, and increases the impact of your data. Next slide. Next slide. So, whoops, I missed it. So, well, that's all right. So one of the challenges we've already talked about is that, an image got disappeared, but in this I had it where they all had different data standards, different data names, different data structures, and that makes the data, you have to harmonize the data afterwards. That harmonization isn't perfect. That harmonization is costly in time and money. So starting off ahead of time again is where you have the biggest benefit and where we can really push the science forward. The other thing that I want to emphasize is that, I think that the biggest benefit is that we can start to get these to use is that NIH is dependent upon peer review, and those peer reviewers are you. And so you can start to look for how data standards and CDEs improve rigor, reproducibility, and transparency in reporting. You should be asking that as you look at papers. Do I understand this? Do they know that that they could compare this data to other sets? And then are they using the best use of that? And I think that all in all, that's going to really help us both as getting your papers hopefully your grants hopefully scored better in review, but also accelerate the translation of your studies. So all in all, I think maximizing the data use is going to maximize the taxpayer investment, which is going to accelerate discovery, and that's really what we're here to do. And we want to, in order to do that, we need to start to adopt the use of these. Next slide, please. But that's going to require a cultural shift, and that cultural shift is starting to be underway. And probably the people who are on these calls are probably not the people who are trying to lead the cultural shift. And so that is to use data standards across sites, across studies, and across disease areas. We've started that. Hiba and Ben and Gabe have started getting that, and Claudio have started getting that done in the preclinical space, and they're continuing to work on it. So, but how do you make a cultural shift? This is the question that we've been asking since we started the preclinical CDEs. And one way, again, is to include it in your study design, in your grant proposals, and budget for it, right? So one of the biggest mistakes I've seen that I know that people were like, I can't afford it, is, well, now guess what? Fortunately, some of the rules around budgeting have been relaxed a little bit, so you can start to budget, hopefully, for the quality of the data. I already talked about incentivizing it through review. So clearer standards should make it easier for peer review to evaluate and make it easier for you to put out the results in papers. It's clearer and it's more transparent. It should enable collaboration. So we did run the Top NT program, which again was to show that preclinical CDEs can use through collaboration. And I know I'm almost done, Gabe, but I only have 2 more points. And the next, the next is, I think probably the most important thing is that we normalize the use of preclinical data elements and data standards through training. Those are the people who are going to make a difference. The trainees are the ones who are going to change the culture and make it the usual to use these things. And finally, just if you don't know, the NIH is working on developing a metric sort of to recognize data sharing as a scholarly output so that you can use this. Hopefully universities and other types of institutions will use this in the promotion, for your promotion, because you're producing quality data that other people want to use, and it's having a bigger impact than your lab alone could have. So with that, I thank you, and I hope that you take this message to heart, that we really need to start. You can be the change agents for preclinical data standards. Thank you.
All right, thanks, Pat, for setting the stage for us. And hello, everyone. My name is Steve Sang. I'm the clinical informatics lead here at NIH Office Data Science Strategy. As Pat mentioned, it's kind of known that CDEs can help facilitate fair research. So today, for the next 15 minutes, I'll share with you briefly some of our efforts and activities in leveraging data standards for not only fair, but I added a couple R's after that, make it more rigorous and reproducible research with a focus on improving the interoperability. Next slide, please. So one of the critical functions of the office is to build strategic partnerships across NIH, and ODSS leads the implementation of the NIH Strategic Plan for Data Science, as shown on the screen. One of the North Stars in the office is to make NIH-funded research fair, as Pat mentioned. Thanks for highlighting this. You can access our plan by using the QR code on the screen. So under this plan, we have 5 different goals, and the clinical informatics effort aligns mostly with goal 2 of the plan to enhance the use of human-derived data, including real-world data from sources like EHR, wearable devices, and so on. The goal, again, is to improve and better integrate clinical and observational data in the biomedical research ecosystem. Next slide, please. Our unit, the Clinical Informatics Unit, promotes the use of clinical standards that enable interoperability. Today, I'm going to talk very briefly about FHIR, and the focus for today's presentation will be in common data elements. For all the these standards, we encourage the use of them through scientific, technical, and operational collaboration with NIH institutes and centers like NINDS. We also work directly with extramural communities, like for me to engage you to present here to promote the use of these standards. The goal is to facilitate the sharing and reuse of clinical and translational biomedical data in research in general. And we do that by leveraging these standards. We are enabling interoperability and improving the fairness ultimately. For all our activities, something we really try to do is to share the resources. So whether from training to all the CDEs that develop in all the projects I will mention in my presentation, we really aim to make them findable, accessible, interoperable, and reusable for the community. Next slide, please. So again, as a premise, very briefly on what is FHIR. Some of you may have heard of Fast Healthcare Interoperability Resources. It basically is a data exchange standard. It's a framework allowing data to be exchanged consistently by providing that framework. It uses standard API. It focuses on how mostly healthcare information can be exchanged among different systems, independent of internal data models or standards. In 2020, ASTP, the Assistant Secretary for Technology Policy, published the final rule for the 21st Century Cures Act, establishing FHIR as the standard required for health IT certification, which in my mind really facilitated the adoption of this standard in the community. Next slide, please. But at that time, a lot of what we saw in terms of FHIR's application in this space, it was really limited. It was mainly for healthcare. So in 2019, we started this FHIR for Research training, focusing on developing FHIR training for the research community with a goal to develop a set of accessible, modular, and reusable training activities. And through this module-based curriculums, users can develop self-directed resources, and including, we included the introductory high-level webinar that's really meant for everyone, and also hands-on workshops and modules allowing specific researchers interested in actually using FHIR as part of their work. Recently, we also added additional modules to train the trainer, allowing this FHIR for training activity to be reused more broadly by the research community. Next slide, please. So FHIR was developed by the community back in 2012, 2013. But really not until very recently, there's been a lot of discussion on preclinical or non-clinical data to be included in FHIR. Genomics was one really good example. This figure was taken from our FHIR training, highlighted that various stakeholders shown as nodes on this picture, and also showed the very complex landscape and modality of data and how FHIR can facilitate the exchange of data in various steps on this picture. So genomics implementation in FHIR is an evolving topic because the complexity of genomic data and the rapid pace of scientific discovery. But this has, I want to highlight this because this is really a very important topic that a lot of people in the FHIR community are discussing. So from this, we kind of expanded on to additional topics like United States core data for interoperability or the trusted exchange framework and kind of common agreement. I welcome you to take a look at the training materials if you're interested. Next slide, please. So every time I give a presentation, I kind of highlighted this as a background. The kind of the reason for us, ODSS, to start a series of CDE-related activities because of this congressional language back in fiscal year 23. We were asked by Congress to look into the use and development of CDEs in research, especially for autoimmune and immune-mediated conditions. So therefore, we started a series of activities I will describe in the next few slides. Next one, please. Inevitably, we will have to kind of tell the community what NIH CDE is. This is the official definition you will find on the NIH CDE repository. It includes a question that tells you what, and it's allowable responses, which tells you why. When you use CDEs systematically across different sites, you ensure consistent data collection, thus enabling interoperability. Next slide, please. So here I have shown an example is what is the patient's height, and the allowable responses could be whole number using centimeters as a unit of measure. So NIH CDEs use this standard terminology concept to ensure that data is unambiguous, it's tied, it's bind, it's tied to usually some kind of vocabulary, medical vocabulary, and shared with the definitions consistent. This reduces confusion and improves communication across different teams and systems. So with this standardized structure, CDEs are machine readable and machine actionable, enabling automation. And CDEs have independent semantics. That means the meaning of the data elements is defined independently of data models or any of the specific forms that uses CDEs. So as a result, these CDEs, these elements can be reused across different data sets or systems, and they can support different allowable responses. So this is the main reason why a CDE supports fair research. Next slide, please. So from the congressional directive, ODSS organized several activities. This is one of them. In this workshop we held in 2024, we discussed the use of CDEs, demonstrate the value of why we need standards such as CDEs. We also talked about how we can implement CDEs and leverage CDEs to make data resources AI ready. So it's kind of to summarize some of the lessons learned from this workshop is we really need to engage the community in developing CDEs. We should not develop CDEs for the community, but we should develop the CDEs with the community. And we should engage not only researchers, but also patient advocates in the entire process. In addition, we should always communicate and collaborate with other people to make sure we understand which CDEs are being developed in the community so we don't reinvent the wheel, and try to reuse those CDEs whenever we can. Next slide, please. Another activity follow up on the CDE workshop is the request for information. So in this request information, we solicited public feedback on the use of CDEs for NIH-funded clinical trials and clinical research, on the technology standard, using CDEs, both upstream in developing CDEs and downstream in implementing CDEs. We also solicited feedback on how NIH CDEs could facilitate research and how we can incentivize a broader CDE usage in research. Next slide, please. This is just to highlight that ODSS, we collaborate across the NIH in various disease areas using CDEs. Obviously, NINDS is an early adopter of CDEs and continues to be a strong collaborator in our efforts in developing and using CDEs in various disease areas. Next slide, please. Here, I'm just highlighting the NIH CDE repository. I welcome you to take a look. The NIH Data Management Sharing Policy supports the use of CDEs to facilitate the consistent data collection. And as noted on the CDE website, that CDE supports fair data principles and increases interoperability for the reasons I mentioned above. And I should also note that they constantly have new features rolled out. Right now, they have a new topics filter, allowing users to more easily find and access the CDEs they're interested in using. So as I noted before, as with all the presentations I give to all the communities, if you're thinking about developing CDEs, this is the first place I would go. Go to the CDE repository, try to see if there's any CDEs in the repository that you are able to reuse. Try to reuse whenever possible. If not, if you cannot find any, try to develop CDEs within your community. And then put them back in the CDE repository, allow other people to reuse them. Next slide, please. So before this presentation, I did a quick look into some relevant CDE categories that you may be interested in. You may be interested to find out there are already many CDEs related to preclinical domains, especially for genomics and various omics, genes, genomic variants and mutations, and also behavior assessments. Here are just a few examples on the broad categories. If you're interested, please take a look at the CDE repository. Next slide, please. So since I joined the office a few years ago, we've been really keenly interested in understanding the landscape of data standards and the use and development of data standards here at NIH. So last year, we started this new effort to use GenAI-assisted landscape analysis when we started to have some access to these systems. So we collaborated with NIAID, the National Institute of Allergy and Infectious Diseases GenAI platform, to do this analysis using retrieval augmented generation, or RAG. So for this analysis, we used publicly accessible data and retrieved funding opportunities from grants.nih, publication data from PubMed, and project award data from NIH Reporter. So we basically look for those artifacts relating to common data elements and then do some analysis on this. Part of this analysis was highlighted on our ODSS director's blog in July 2025. You can find that blog using the link on the slide. So since then, we have expanded this analysis, not only focusing on CDEs, but focusing on FHIR. And also we continue to kind of expand our effort from CDEs into other domains, such as ontologies, semantic similarity, and knowledge graphs. So this is one way for us to understand what kind of impact we are receiving from our various data standards efforts. Next slide, please. So with that, I just wanted to acknowledge all the team members from the ODSS Clinical Informatics Unit. And then I think this is my time to hand it over to Dr. Carolina Mendoza Puccini for her part of the presentation.
Okay, thank you, Steve. So I'm Carolina Mendoza Puccini. I lead the NINDS Common Data Element. So before I just said CDEs, now I have to specify that they're clinical because we didn't have for many years preclinical. Next, please. So you already heard from Steve the definition of CDEs. So CDEs is more than only a well-defined variable with a question and an answer. So it has so many different additional information that makes it unique and unambiguous. And I'm not going to go through everything because Steve, I think he shared the link to the workshop. And my colleague, the next two slides are from my colleague Denise Warcel from NCI, who has an excellent presentation on what entails a good CDE. What does it mean to be a good CDE? Like to have a standardized structure, standard terminology concepts, the independent semantics, unique identifiers, and also the compliance with the FAIR principles. Next, please. So I think one of the main goals is interoperability. We already heard that from all the presenters before. And that's the ability to seamlessly and efficiently exchange or reuse data with clear, unambiguous meaning. So that's like the key word there. So there are principles of interoperability, the standards for exchange, rich metadata, shared semantic alignment and mapping, and good governance among the stakeholders. And this applies to both CDE and data interoperability. And I encourage you to go and look at those workshop videos because they're excellent. Next, please. So now let's jump, because this is like a very brief overview, to where do we keep most, but not all, of the NIH CDE most of the CDEs at NIH, sorry. So we kept them at the NIH CDE repository, and this is hosted by the National Library of Medicine. Next, please. So the CDEs, we have two types of CDEs, and we're gonna talk later about that in the repository. We have a group of CDEs that are endorsed by a governance committee, and most of the CDEs that are not. So what does it mean? The NIH Scientific Data Council assigned a group of people, and I co-chaired this group with Ken Wilkins. And some of my, I think one or two of the members are connected today. And they tasked us to review CDEs to make sure that they're of excellent quality and then endorse them. And they gave us, okay, you have to follow 5 criteria. The first one is that it has a clear definition, or the variable has a clear definition, measure with prompt and response. And Steve talked about that definition. Also that there's documented evidence of reliability and validity. We prefer human and machine readable format. I think all of the ones that we reviewed are meet this criteria. And also that they're recommended or designated by a recognized NIH body. So this means that whoever submits the CDEs have to be working with someone from NIH, either a program officer, program director, or anyone at NIH that can be like the point of contact. And then the last one, which is very important, is a clear licensing and intellectual property status. So obviously, the ideal is that everything's open source, that we don't have to pay. But in some cases, the gold standard to assess something, you have to pay for it. Next, please. So these slides are from my colleague, Robin Taylor. Shout out to her, because she leads all the QA at NLM. So the first step is, and we provide all the documentation for people to prepare the submission. So we provide a guidance document, the submission form, and then a submission form with an exemplary of what we mean and with their own definitions. So it starts with, this team of people putting together all the CDEs. And one thing that I want to clarify is that we don't assess scientific merit. We only focus on the CDE quality. The scientific merit is because it comes from a group of experts, and we ask for that information too. So first thing is submit the CDEs, then it comes to us, we usually accept them, and then the third step is it goes to a QA process, which is lengthy and, well, not lengthy, but it's comprehensive. And that's done by the National Library of Medicine, the QA team, which, they're all excellent. Then they may have some back and forth with the submitter just to address gap, make sure that nothing is missing, there's errors or anything that they may have questions on. So once everything is clear, all the queries addressed, they go to the governance committee. So we have, we select from the committee a group of people that will review the CDs, making sure that the criteria are met. We usually have comments, it's like any other review that you have. And then we may have some just minor comments, and then they're ready for endorsement. It depends also on the number of CDs submitted. Sometimes, it's a very small collection of 15 or 20, and they don't have a lot, so it's easier. But sometimes we get hundreds. It can be, we may have to break it into like 150 per review or 100, depending on the complexity. And then once and we send a report to the submitter. Once everything is addressed, it comes to us, we review it, and we may have to meet again, or we may not. And then we endorse them, and they're at the end published as endorsed in the repository. So I'll show you, and I think at the beginning we had like the golden ribbon, but next, please. So this is the link and some information. Steve also showed something similar. That's for you to know, because we're going to share the slides. Next, please. So that's the golden ribbon there. Means that the NIH Governance Committee endorsed these CDs. So we currently have 177 endorsed CDs in 4 different collections. But as I mentioned before, we also have other CDs in the repository, and they're about 22,000, and they come from different NIH institute centers, offices. And we also have 1,600 forms. And I'll provide more information on, details on what I'm talking about at the end, so you can go and visit the NLM page. We currently have more than 1,000 CDs in the pipeline to review. So this year for the governance committee will be very, very busy, and we hope to, to have have them all reviewed at the end of the year, and they will be posted. And then, the good news for the preclinical world is that the first preclinical CDEs that will become NIH endorsed in the CD repository will be from the NINDS NT Proceeds program. And Hiba is going to talk to you about that, which is, super exciting. It's the first time that we're going to have preclinical CDEs. Next, please. So I'm not going to go into detail into this. This is just a list. So to illustrate that we have many of the institutes at NIH are using CDs or are developing CDs. We have many of them that are in the process of developing in new disease areas, like the autoimmune, and Steve mentioned that in the in his presentation, and then we have many others using. NCI was one of the first institutes that started using CDs, and they have also an excellent page with a lot of information. Okay, next, please. So at NINDS, or NIH in general, and Pat already showed something like Fitbit, we have a TBI specific thing. We have NIMH's mental health. They have their own CDs at their own repository. And, as I mentioned, NCI and other institutes. But this is just a very small example. And also the HEAL CDs that I don't know why I didn't put them here because I direct that program. But there's also a fantastic resource for paying CDEs. Next, please. And at NINDS, we've been in business for a long time too. I think we were probably after NCI, the next. And the first ones developing CDs in neurological disorders. So we have like 23 disorders. Many of them have pediatric CDEs. We recently published the mitochondrial, Parkinson's, and the TBI version 3, it's coming. And this is all clinical. Next, please. Also, like a representation, very busy slide, but it's just to show you that at NINDS, we have different networks, clinical networks, so StrokeNet, NeuroNet, which is already completed, but then Siren took over that, the emergency part. They all use CDs. These are very large clinical trials that have thousands of participants. So the use of the variables and CDs is like in the millions in terms of how many participants. And we also have intramural, also uses CDs. We have other network studies and trials. Next, please. So this is the process. So this is to show you that developing CDEs is not something that you do overnight. It takes a lot of planning, a lot of time. And this is only from the time we invite the members. So depending on how large and complex is your project, it will depend on how challenging it will be to select the number of people that you want in the table reviewing this, and I'll go into detail. So for clinical at NINDS, I can only speak for my project, is about 18 months from the time we invite the members to the time we post them. So it goes through a lot of, internal reviews, the actual meetings where they select everything, internal reviews, public review, and then the posting in the website. And it's a continuous process. We review this every year as much as we can, and if we have the funding. Next, please. Okay, so this is a scenario, and this is also from Denise, that we have to take into account when we develop the CDEs. So first you have to decide, do we want to use CDEs? And then, that will depend on the funding agency, regulatory agency, if it's a professional society, etc. Then choose the CDEs. So how are we going to do this? And I think something that is not here is the reuse, identify, are there already CDEs that exist? And then the third point will be who, is this, like if it's a funding agency, do you already have CDEs that they require? Is it mandatory or like you can choose not to use them? And if there's how do you monitor and assess the compliance? How do you enforce the use of CDEs? Next. And I think this one is one of the most important things, and I can tell you what were my pains, so I can share with you. And first, you have to take into account there is a learning curve, and Pat mentioned that. Adoption is hard. It is slow. It's stepwise. There's going to be a lot of pushback, but I think you have to be persistent. When I started this project 9 years ago. 9 years ago, many people didn't even know what data standards were. Now I get a lot of calls of people, can you develop, in this disease area? There are many, many people interested in developing or trying to get some advice if they want to do it themselves. Also, coordination, and Pat also mentioned this, do not reinvent the wheel and do not work in silos. And I cannot stress this enough. Try to get all the stakeholders, not only from academic institutions, nonprofits, industry, regulatory agencies, like everyone you think that can contribute or may use the CDEs or may benefit from the use of CDEs. Contact them, talk to them, get together. Also, another thing to take into account, I know that many of you have collected data for decades probably, and you already have a way to collect that data. So it may be a little challenging, but not impossible to retrofit or map that data to any new CDs that you are going to develop. And again, let me repeat this again, like 100 times. Try to do it from the beginning. Do it when you're planning your project. Do you already have tried to identify reuse? And then after that, if they're existing, they don't exist, how do we develop? And contact other people that have done it before. Next. And I think this is it. I also have, some limitations. And the main one, I would say, don't work in silos. Don't have different groups working on the same thing, because it's a waste of resources. And I think next. Thank you. I will, we'll share these slides. They have all the information. They have additional information with other links. And thank you so much.
All right, thank you to all of our speakers. I'd like to invite our other speakers, Pat and Steve, if you can come on camera so that we can start our panel discussion. I'd like to invite our audience to please put your questions in the chat. You can also use the raise hand option, and we won't be able to put you on camera, but we can unmute you to be able to participate. But I do want to give a round of applause for our speakers. I think that was a wonderful, very comprehensive round of presentations that showed us, the importance of this cultural change in the preclinical world that we're trying to address with the NT Proceeds program. And it is a partnership with our, partners from other federal agencies, including the DoD and the VA. And of course, we have 3 communities that are part of that program. So we have the traumatic brain injury community, which are presented by Precise, which is our interagency resource center for preclinical TBI research. And then we have the International League Against Epilepsy, as well as the American Epilepsy Society Task 3, which are representing the post-traumatic epilepsy community. And then we have our spinal cord injury, and we have multiple spinal cord injury foundations, which we'll put on the last slide, but I don't want to miss anybody. So we're very grateful for this new program, and we're currently, as we mentioned in the first webinar, in the process of developing those CDEs, and we anticipate that in April we will be able to have a webinar to start showing all of these CDEs that we've been working on in the background. But I don't see any questions in the chat, but I do see some raised hands. If we can have our first person, I can't tell who has their hands raised. Let me see.
I can go ahead. Looks like we've got Sanjoy Bhattacharya. I can go ahead and
Yeah,
allow.
Dr. Bhattacharya.
Hello, can you hear me?
Yes, yeah, go ahead.
Okay, so I have two things to say here, really. One is that the CDE is not standardization. The two are two different things. Standardization in experiments happens separately. That should be encouraged. The CDE is an approximation, but not standardization. And the second thing that I would say is that you guys should do something to socialize. And part of that, I would recommend that post meeting, those who have at least registered, if you send these slides, all presentation slides, and I wonder if you can encourage all the people who are registered to share it in their LinkedIn or something like that so that this is socialized. I think these CDs are very incredibly important thing. Even if people do not adopt, they should actually consider and they should be aware of that. I do not think in universities you have wide adoption. And these are the approaches. Thank you.
Thank you for the feedback. And yes, we are counting on our community members to help us with spreading the word. So we'll make sure to reach out where possible. Do any of our speakers have any specific comments on how CDEs are standardized versus not, or can we move on to the next question?
I mean, I think the point is taken, and didn't. What we need to do is make sure that CDE, in my perspective, CDEs, the component CDEs provide is transparency in reporting and standard in reporting variables in the same way. So whether you want to call that a standardized reporting or not, I don't know, but that way it's much more easy to reproduce because you know which factors you need to do, and so. Yeah, socializing it, as we said, socializing it and getting the cultural change has been something we've been talking about for 15 years, and it's a tough, right? People have been collecting data the same way for a long time, which is why what I know I've saw is the next generation is really the generation who's going to be more easily to adopt it, but it's hard to change when you're a gray hair like myself.
Thank you. And yes, we are posting all the recordings onto our YouTube page. And so people should be able to access all the slides. All right, Ramesh. Dr.
Thank you.
Ramesh.
This is very, very helpful. One question I've had is, how do we, as people who are implementing CDEs and kind of, how does this then
Hello. Yes.
translate to our grant applications? Like, what do we put in grant
Yes, please,
applications?
Karim.
And how are study sections and peer review panels going to view information about these CDEs in grant applications?
So I'm the one that mentioned it. Hi, Ramesh.
Yeah.
So, so the point is that there is no requirement that you use CDEs. It's not a review criterion. My point was simply that as a reviewer, um, it, I'm hoping that it would help you understand better what they're trying to do because there'd be more transparency and you could look at the CDEs for the measures they're doing. I think you can refer to, so what Precise and the NT Precedes process are doing are going to provide you, I think NT Precedes is or Precise. Anyway, they're going to come out with papers in which you could reference them just to say we're going to use the standards based in these, or the CDEs based in these papers. And here's the data dictionary. So Precise will be hosting the data dictionary, and so I think that the more it comes in my view, in the grants that I've reviewed before I was in this job, I think a CDE would have made it easier to understand exactly what they're measuring, which to me makes my job easier as a reviewer.
Great. Thank you.
Thank you. I don't see any additional questions, so I'll ask a question. So Steve, So you mentioned that the genomics are using, the FHIR for data standards, but I wasn't very clear as to whether they have developed CDEs, because we do have a genomics working group that is developing CDEs in the NT Proceeds program. And if you can speak more to that and how we can have that translation between the preclinical and the clinical data standards that we talk about, especially where it comes to CDEs.
Yeah, thank you. Thank you for that question. So FHIR, by definition, is not a NIH CDE. In fact, when I presented as a framework or data exchange standard. Simply put, FHIR has the question, but doesn't have the answer. So it has the question, what is the patient's height? But it's flexible that anyone can use really any answer. So it's not by NIH definition what a common data element is. That being said, the problem in the FHIR community is FHIR was never considered as part of the FHIR framework. But now, because of genomic testing of the variants relating to various diseases, we started to see that historically more clinical community is interested in adopting this preclinical, almost data element or domain. I think that's an encouraging sign that when I say biomedical research and the clinical research communities are converging, which they should, because there must be some kind of translation that's happening. And not only the data, the research, but somehow how the data is exchanged. So that's the importance of the FHIR community kind of considering, including at least discussing how genomics data should be handled within the FHIR framework.
Thank you. Yeah, so we'll make sure that our genomics working group will, reach out to the current working groups and hopefully all the resources that we've shared with everyone, people can go for the training. And I guess another question for Carolina. So we know from our experience with the NT Proceeds program and other preclinical CDE efforts with NINDS that there is a challenge in some of the terminology between the clinical CDEs and the preclinical CDEs. So for example, what you guys showed in your presentations as the preferred question text. When we're working with animals, we don't really ask the same questions as when we work with participants that are human subjects, So, so if you can speak a little bit to the flexibility of, the language that is used when defining a CDE, and how the NIH endorsed governance committee sees that.
Well, remember that we're new to the preclinical too, but we definitely have flexibilities. We're not going to pretend to collect data or require fields that don't apply to preclinical. So we'll work with you. That is not my area of expertise, in terms of the data science part. That's why, I have a lot of data scientists, or we have in the governance committee. But when we review, we take in account that. I know that we've met, with, I don't remember what group that was doing preclinical. Was this one? And we talked about that. So we are flexible in terms of that. Obviously, preclinical is so hard. And when we designed and developed the submission form, honestly, 100%, we were thinking about clinical. So we may have to rethink or revisit the form and maybe create one for preclinical. But I think that so far with your submission, it worked. So I think it will be case by case scenario.
Yeah.
And we may have to bring maybe you, Hiba, someone from the preclinical world that developed these CDEs and say, okay, what do you think that we should change, in these forms, and then adapt it to the preclinical.
Yeah.
But we'll work with you. That's the point. We want to endorse the CDEs.
Excellent. We're so happy to hear that. And so for the sake of time, we are already past 3 o'clock. We do have some questions coming in, and I see that our panelists are trying to address them as they go through. This is still just the beginning of the conversation. We will have a lot more webinars to keep addressing all of the challenges that we face in the preclinical space and to update the community on what's happening. Gabe, if you can move to the next slide, please. We would like to invite everybody to, thank our speakers. So a big round of applause for our speakers today. Our next webinar will be in March, so look out for an email. We'll be sending it to all of the registrants, and we also post the information to our website. We do have a lot more topics coming up. Our next topic will be covering repositories. So stay tuned, and we look forward to everyone's participation. And if you can show the idea scale QR code, let's continue the conversation on our Idea Scale community platform. It should be very easy for you to be able to log in. And then once you're logged in, you can post your ideas and your comments, and we can continue the conversation offline until we come to our next webinar. So thank you all for attending today, and thank you for our speakers, and for Gabe and Ben and Claudio for co-organizing with me. Thank you. Have a good day.